In this video, we're going to talk about some of the common basic structures of prokaryotes. In a previous biology class, you should have learned more about cells. Now we have two kind of major categories of cells. We have prokaryotes, or prokaryotic cells, which is what is being shown here. And then we have eukaryotic cells, which is being shown here and here. And this more specifically is an animal eukaryotic cell. This is a plant eukaryotic cell. There are also fungi or fungal eukaryotic cells and protists that fit in this eukaryotic category as well. Now, as a reminder, all cells, regardless of whether they are prokaryotic or eukaryotic, have these four structures. So they all have a cell membrane, DNA, which is the genetic material, ribosomes, which make proteins, and cytosol, which is the liquid goo inside the cell, which is also oftentimes called cytoplasm. So you can see in these pictures here, I've kind of circled some of these structures, but let's take another look at them. So on our prokaryotic cell down here, this would be a bacterium. And you can see that it has a cell membrane. It has the ribosomes. It has DNA, which is this material in the middle here. And it also has cytosol, which isn't really shown in this, this image here, but the cytosol would just be all this goo kind of inside the cell. Our animal cell, you can see that it has chromatin, and chromatin is just DNA that is packaged in or packaged around proteins. We have ribosomes, which are either free-floating or they might be attached to the endoplasmic reticulum. We have the plasma membrane, which is the cell membrane. And then we also have the cytosol or cytoplasm, which once it, actually it is here, right here. So the cytoplasm is just this goo that kind of fills up the cell. It's inside the cell membrane, but outside the nucleus. Plant cells, we also have that plasma membrane, which is our cell membrane. We've got chromatin, which is our DNA, once again, wrapped around proteins. Our ribosomes. And then there's also cytosol here, which I don't believe it's actually labeled on this picture. But once again, the cytosol is just going to be this goo that kind of fills in the gaps in the cell. Now that we've covered the four structures that are found in all cells, let's take a look at some of the major differences between prokaryotic cells and eukaryotic cells. So the main difference is that prokaryotic cells do not have a nucleus that is surrounded by a membrane. So remember that prokaryotes do have DNA, but that DNA is housed or kind of packaged in the nucleoid region. And this is just in the cytosol. So there's no membrane surrounding that DNA. Eukaryotes do have a membrane-bound nucleus, and that's what this kind of purple structure is here on the animal cell and the plant cell. Prokaryotic cells also do not have other membrane-bound organelles, so they do not have things like mitochondria, endoplasmic reticuli, Golgi apparatus, etc., whereas eukaryotic cells do have that. Prokaryotic cells are also smaller than eukaryotic cells, about maybe 1/10 the size, so significantly smaller, even though I did not put these pictures to scale here. And prokaryotes are all unicellular organisms, so they are one cell makes up the organism. Now, they do sometimes form colonies, which are multiple cells that kind of hang out together but they're still operating as individual organisms even as they exist in these colonies. Now, organisms made up of eukaryotic cells might be unicellular, so they might be single-celled, which is the case with protists, or they can be multicellular, so plants, animals, and fungi are all multicellular. So eukaryotes can be either unicellular or multicellular, whereas prokaryotes are only unicellular organisms. Now, these are just some of the main differences. There are additional differences between prokaryotic and eukaryotic cells, and we're going to focus on some specialized structures that are only associated with prokaryotic cells on the next several slides as a way to kind of point out these specialized structures and their functions. So the next several slides are going to talk about what we call kind of this typical prokaryotic structure. But prokaryotes, just like eukaryotic cells, can come in different shapes and sizes and different varieties. So we're going to look at kind of the general idea of what one looks like, but know that there's actually a lot of variety in real life. So first, let's take a look at the genetic material. So as we mentioned before, prokaryotes do have DNA. Their genome, and remember that the genome is just all the genetic material so basically all of the DNA needed to make an organism is kind of encompassed in the genome. So all of the genetic material needed in this particular prokaryote is going to be packaged in a single circular chromosome. Now, by contrast, if we were talking about eukaryotic cells, they typically have multiple chromosomes. And their chromosomes are bar-shaped or linear. So it's kind of hard to see in this picture because it's got the DNA kind of all wrapped up around itself. But if we were to take this DNA out and kind of stretch it out, it would ultimately make one circular loop of DNA. So remember that prokaryotic cells do not have a membrane-bound nucleus like eukaryotic cells do. Instead, that chromosome is located in the nucleoid region of the cytosol. So you can see that here, this is considered the nucleoid region, which contains the DNA, which is packaged as a single circular chromosome. Now, one interesting structure that some prokaryotes have that eukaryotes do not have are these things called plasmids. And these are extra rings of DNA. So this single circular chromosome is really all the bacterium or prokaryote needs in order to survive. But there can be extra little rings of DNA out here in the cytosol. And these plasmids, they're not necessary for survival, but they might give the prokaryote an advantage of some sort. So for example, we're going to talk about antibiotic resistance later on. And antibiotic resistance can be linked to genes that are found on these plasmids. So if the bacterium has the plasmid with this little extra gene for antibiotic resistance, then it might be able to survive in cases where other prokaryotic cells without that plasmid are not able to survive. So these are extra rings of DNA that are not part of the main genome, and they're not technically necessary for survival, but they might give the prokaryote some sort of advantage. Since bacteria and archaea are both prokaryotes, meaning that they are both cells, it means that they both also have a cell membrane, which is often also called the plasma membrane. Now, there are some differences between the cell membrane of a bacterium and an archaea. So with bacteria, their cell membrane is made up of a phospholipid bilayer. And if you look at this picture over here, this is kind of what a phospholipid looks like. Sometimes we also draw it just kind of like with a little head and little tails sticking off of it. You've maybe seen it drawn as a bilayer with the heads on the outside and the tails kind of sticking in the middle. So a phospholipid bilayer means that you actually have two layers. So this would actually be two layers of these phospholipids that are turned in towards each other. So bacteria have a cell membrane that is similar in structure to a eukaryotic cell in that it is made up of two layers of phospholipids. Now, archaea can have a phospholipid bilayer, or sometimes they might just have a monolayer. And this just means that they have one layer of these phospholipids with their little tails sticking out, but they do not have that second layer that is sandwiched together with them. Now, both bacteria and archaea have a phosphate head, which is shown in this kind of green area right here. And this is the same thing that we would see in a eukaryotic cell as well. But the lipid tails, so this area here, which is also shown in these little tails here, they can vary in their molecular composition. So with bacteria, their lipid tails are made up of fatty acids. So this is similar to the phospholipids that make up a eukaryotic cell membrane. Now in archaea, you can see that the lipid tails look a little bit different. So you can see with the bacterium here, the lipid tails are forming these little zigzags here. And these zigzags are just representing carbons that are actually bonded with some hydrogens. Now, if we look at the archaea up here, you can see that their lipid tails look a little bit different. They have these little extra groups of carbon attached to them. And so the lipid tail actually contains isoprene, which is also known as phytanil. Prokaryotic cells also typically have a cell wall, and the cell wall has these functions of protecting the cell from outside threats. It also helps to determine the shape. So these are three kind of common shapes that bacteria are found in. So cocci, singular would be coccus, is a spherical shape like what we see in this first picture. Bacilli, or singular is bacillus, is what is shown in the second picture. And then spirilli, or spirillus, is shown in this third picture here. Now you may be familiar with the cell wall of plants because plant cells also have a cell wall. But in plants, the cell wall is made out of cellulose, which is a polysaccharide. It's also more commonly referred to as fiber. So when you are trying to get fiber in your diet, it basically just means that you're trying to eat more plant material. Now, the cell walls of prokaryotes differ from the plant cell walls in their structure, and they differ from each other. So in bacteria, the cell wall is made of peptidoglycan, and this is a molecule that is made up of polysaccharides, which remember polysaccharides are a carbohydrate. So a polysaccharide is just a chain of monosaccharides. And peptides, remember peptides are related to amino acids and proteins. So the cell wall of a bacterium is essentially made up of proteins that are bonded to carbohydrates. Now, bacteria, you've maybe heard the term gram-positive or gram-negative, and we're going to talk a little bit more about that on the next couple of slides. But whether a bacterium is gram-positive or gram-negative partially has to do with their composition and thickness of their cell wall. Now, archaea also have a cell wall, but their cell wall can come in a variety of different forms. So their cell wall might be made up of pseudo-peptidoglycan, which is similar to peptidoglycan, but they're different sugars. So the carbohydrate here that's found in bacteria is going to be different in the archaea. It might be made up of polysaccharides. So this is just a carbohydrate, so just a whole bunch of monosaccharides bonded together. It might be made up of glycoproteins, which are proteins with carbohydrate chains attached, which might sound like they're the same thing as a peptidoglycan, but they differ in how the proteins and carbohydrates are bonded together. Or the archaean cell wall might be made up of just a protein. So as we mentioned on the previous slide, the main difference between gram-positive and gram-negative bacteria is the composition of their cell wall. But really, the difference is what we can see, and it has to do with the special staining technique that was developed by Hans Christian Gram. So the Gram actually comes from the scientist's last name who developed this staining technique. Now in this picture here, we are looking at some different types of bacteria. The ones that are darker purple are gram-positive, and the ones that are a lighter pink color are gram-negative. So we can visually see the difference between gram-positive and gram-negative bacteria when we are looking at it through a microscope. Now, if we analyze the molecular composition, we can see differences as well. So gram-positive bacteria typically have a thicker cell wall that is made up of 90% peptidoglycan. Remember, peptidoglycan is that molecule that consists of polysaccharides bonded to amino acids or proteins. Gram-positive bacteria do not have an outer membrane, and as we mentioned, it stains purple. Now, gram-negative bacteria have a thinner cell wall, and only 10% of it is made up of that peptidoglycan. It also has an outer membrane that surrounds the cell wall, and that outer membrane might consist of lipopolysaccharides. And if we break down that word, lipo just means lipid, which means a type of fat, and then polysaccharides are carbohydrates. So this is just a molecule that consists of a complex carbohydrate somehow bonded to a lipid or a fat. There may also be lipoproteins, and once again, this is just a protein that is somehow bonded to a lipid molecule. And the gram-negative bacteria stain pink when you look at them through a microscope. So that's what we're seeing kind of in here. This drawing helps to visualize the differences in the cell walls and outer membranes of gram-positive versus gram-negative bacteria. So on the left, we have our gram-positive bacteria, and on the right, we have our gram-negative bacteria. Now you'll notice that from about halfway down, they are the same. All right, so you can see that this inside portion here is the cytoplasm. This is also the cytoplasm in this bacterium here. They have a plasma membrane made up of phospholipids. And proteins. So the main difference happens outside of the cell membrane. So first let's take a look at this peptidoglycan cell wall. So you can see in the gram-positive bacterium that the cell wall is thicker, whereas in the gram-negative, it is thinner. And there's just less peptidoglycan in the gram-negative than there is in the gram-positive. You can also see that the gram-negative bacteria has this outer membrane here that consists of those lipopolysaccharides and lipoproteins that we talked about on the previous slide. Some prokaryotes also have a capsule, and a capsule is the outermost protective layer of the cell. So if we were to kind of draw this prokaryotic cell like an onion, we would have the cell membrane, and then the cell wall, and then the capsid would be outside of, the capsule would be outside of both of those. Now the capsule helps to provide additional protection to the prokaryote, so it helps to prevent from dehydration. It also helps to avoid phagocytic cells, and phage, or phago, means to eat. And these phagocytic cells are often associated with the immune system. And so this capsule can sometimes help this bacterium evade the immune system of, potentially a host that it is trying to make sick. Some prokaryotes have a flagellum. So flagella is plural. Flagellum is actually singular, and flagellum is just this kind of cellular extension. It's usually a protein extension that's coming out of the cell, and it's typically able to whip around like a corkscrew, and it helps to allow the prokaryote to be able to swim or move around in its environment. One more structure that is oftentimes associated with prokaryotic cells are these things called pili, and singular would be a pilus. And you can see it here on this picture. There are these little cellular extensions that are sticking out of the cell here. Now, the structure and function of the pili is different from the flagellum. So primarily the function of these pili, it might help the prokaryote attach to a surface. It also plays a reproductive role, and we're going to talk about a form of reproduction in another video called conjugation. And you can see that in this picture down here. These are actually two different bacteria that have connected themselves to each other using this pilus. And in doing so, they're actually able to exchange genetic material with each other.